Let me get rid of the camera also. There you go.
All right, thank you. Thank you, everyone. Can you hear me okay in the back? So good afternoon, everyone. And to those of you online, thank you for being here for my presentation today, which is a really long title, purposefully in a sense, so you have to think about it. Shooting for the middle misses the mark, repurposing real world data to improve drug dosing. And I thought I'd start with kind of giving you a little bit about myself for the students maybe out there that don't know who I am. I've kind of had a sort of a nonlinear path to where I am today. And I started off, I'm East Indian by origin, born and raised in Africa, Southern Africa. And I moved to the United States in 1992. And when I first came here, I was in community college for 2 years. So for students out there that are thinking about a path, you can make it with a community college education. And from that, I moved to University of Texas, got my PharmD there. And one of my mentors there sort of encouraged me to consider an academic path. And I had two choices at that point in time, either pursue a PhD or pursue a residency and fellowship path. And I chose the residency and fellowship path because I wanted to be more of a clinical scientist. So that was the path I took, and I went to upstate New York, completed a PGY1 residency, and that's actually when I got my first taste of specific population pharmacokinetics. I was sort of thrown into this study where we had to collect samples from patients that were obese in the OR undergoing bariatric surgery. So that was sort of my first introduction to that space. From there, I completed a fellowship and took my first faculty position at the University of New Mexico. And really, from that point on, I've moved quite a bit, but the big transition for me was leaving academia. I left academia and I joined pharma for about 6 months. And it was during that experience what I think of as a game changer, because I really got a deeper understanding of the roles that academia, industry, and other groups have in that drug development process. I went back to academia, and as you know, I'm now at the University of Michigan and have gone through that promotion process and I currently serve as chair. So what I'd like to do today is really start off with the problem. When we're thinking about drug development, we think of these standard phases, and we often think of them as sort of a linear path. For those of you who've been in drug development, it's actually not a linear path. But typically, when we're thinking about the populations that we study healthy individuals where we do a dose escalation, try and get some sense of the safety of the compound. And then we try and get a sense of whether this compound has any effect. And that's what phase 2 is. From that information, we build models. We get some sort of prediction of what that dose should be and try and get the best guess using population PK modeling of what that dose should be in phase 3, because we want to get it right. We want to have a dose that's effective and safe. But once that's done, we have to use it in everybody else. And that everybody else can include obese individuals, underweight individuals, and we often leave behind a lot of underrepresented populations in that drug development process for various reasons. It's expensive to do, it's risky to do. So for example, pregnant patients, we can't really include them in the early designs for that reason, because we don't know anything about that compound, right? So the young, the really old, pregnant patients are some examples of the specific populations that we don't know. And so we're really reliant on phase 4. This is after the drug's approved to try and figure out what that right dose should be in that population. And that's kind of what I'm going to focus on today is that box, which is how do we scale that information to individuals that are overweight and obese. And the big gap, again, is recognizing that the populations that we study are very different from the populations that we actually treat. So why this is relevant is because obesity has been increasing in prevalence for the last 50 years. The current projections are that 50% of adults will meet the definition of obesity by 2030. And the portion of individuals that have a morbid obesity, which is a body mass index greater than 35, that I'll talk more about, is going to be close to 20 to 25% of the population by 2030. So it's a really, really big problem. Why this is a problem from a dosing perspective is because of the way in which we scale this information. The standard model has been to think of individuals as sort of this reference average person. That reference average person is a 70 kilogram individual with 1.73 meters squared as the basis. And this has sort of been the physiologic standard since the early 1920s. If we look at our data now in the United States, what you see is that the average adult is closer to 90 kilograms, or about 200 pounds, or 2.1 meters squared. And the average woman is 77 kilograms, about 1.86 meters squared. So why this is relevant is that when you do your projections and you project to that 70 kilogram individual and you come up with a dosing regimen, often you're likely to be underestimating that average by about 10 to 30%. So there's a lot of source of error when we scale to that middle. Now, what I'm going to do is walk through an example. This is a gene therapy. So what do I know about gene therapy? Not a lot. This is not my area of knowledge, but I'm taking this case example just to illustrate the way in which we approach drug dosing in development. So this drug was approved in 2022, November. It is used, it's a gene therapy to treat hemophilia B, which is a rare disease and involves non-production of factor IX, which leads to this risk for bleeding in males because it's an X-linked genetic disease. So this is a typical package insert from which we think of as pharmacists what the dosage should be. And what I'm focusing on, again, is the dosage and administration of this gene therapy and potentially its use in these specific populations. This is the standard approach by which information is relayed to pharmacy when a drug is approved, right? So this is the case for Hemgenix, and this is directly from the product label. So this gene therapy is delivered as 20 trillion gene copies per kilogram of body weight. And so to make it easy on the pharmacist, they have given you this dosing calculation, which is basically take the person's weight, multiplied by 2, that's the dose in ml, and they give you an example. That example is a 72 kilogram individual. And you say to yourself, well, why didn't they just use a 70 kilogram individual? Well, the reason they used a 72 kilogram individual was it's an illustrative example to the pharmacist that if you calculate the dose and that dose exceeds the vial size, go to the next largest vial. So in this scenario, the person would have calculated, you know, it's 144 mLs. If I had 140 mLs, I would have used 14 vials, but because it's 144 mLs, I'm going to use 15 vials. So why am I using this example? Well, here's a typical scenario again, no information about the use of, obviously this is, I said, an X-linked factor. So, you know, we don't expect to use it in pregnancy. It's unlikely we're going to understand what it's used in pediatric patients, but in that geriatric population, they say we studied 6 people. We don't think there's a dose adjustment. That's the basis. But here's the real reason why I'm talking about it. This gene therapy, on average, costs $3.5 million. This is the most expensive drug that we have on the market today. Okay, is this exactly what the cost will be for an individual? I don't think so. Most people will not be able to afford this. So the true cost will be different. But again, illustrative example, 70 kilogram individual, the cost is $3.2 million. Each vial costs what I consider the cost of a house. It's about $300,000 per vial. So if I'm computing 14 vials and just slightly over that, it's an extra $300,000 per dose, right? But in this package insert, they do give you, there's not one mention of the word obesity. There's not one mention of any of these things in the New England Journal of Medicine article. But in the label's written out all the way to an example of a 120 kilogram individual. So clearly they had data in obese individuals. And the point being is, for all these projections, you're basing it on this average individual. And fundamentally, do we really need higher doses in these bigger individuals? Well, the package insert doesn't share that information. But if you're a clinical pharmacologist, what you're going to do is look at the briefing document. So what is not known to most in the public is that there's a lot more information that goes into these kinds of decisions. And there's a lot of information that's perhaps not public. But the FDA asks these kinds of questions clearly. And if you look at the briefing document, what you see is the factor 9, which is the determination of the activity of this gene therapy, actually was higher in overweight individuals. Obese individuals had a higher response to this gene therapy. So what does that mean? It means that either the obese individuals got the right dose and everybody else is not getting the right dose, or they're getting too much, right? An issue not addressed for this gene therapy. And this is, again, one example of many examples that we have when we're thinking about drug dosing. So fundamentally, should larger individuals get bigger doses? You know, if you go and you say no, I think the answer is no. Well, that's correct too. So the answer to this question is really, it depends, like everything else, right? So correct, the exposure response for a lot of drugs are not dependent on weight. And if you say yes, then that's also correct. The exposure response for some drugs will correlate to weight. But how should we do this? If we determine that there is a relationship to body weight, how should we do this? Well, there's two ways to think of this. One way is to think of it as a process that's linear and proportionate. And the other way is that it's nonlinear or not proportionate. And from my experience and my understanding over the last 20 plus years is that in most cases, that model of nonlinearity is true when we're thinking about body weight. And the problem is perspective. If I only study this group of individuals here, I could fit this information as a linear function or a nonlinear function because it's close enough mathematically. If I focus in that middle box, which is typically what happens in drug development, all three paradigms could exist. I could argue for a fixed dose because I could say that clearance is not changing enough. I could say it's weight-based, or I could say it's something else. And so all those paradigms could exist. And it's because of, again, perspective. So the paradigm often is, for the modeler and for the company and the process, is you must choose wisely. What are those choices? The choices are most often fixed dose. And that's reasonable. Often it's the formulation that's driving that decision rather than the correct decision of the dose driving the formulation, right? Think about it. If I could just make one formulation that's 500 milligrams and use that for as many people as possible, that's going to be the simplest option. That's going to be the development pathway. If I have to make fractionations of those doses, that's a less likely pathway. The second pathway is weight-based. We employ this a lot for intravenous compounds because that's a strategy that we think achieves precision. And then the third option, which is a process by which we deliver cancer chemotherapy, and that's body surface area based. So those are really the three big paradigms that we currently employ. And so why body surface area? So weight is something that most of us can understand, and fixed dose is something we can understand, but why body surface area? And the origins of body surface area are old. They start off, in 1870 as just a determination of trying to compute what the area is for many other physiologic reasons. And so body surface area is mathematically just a function of 2/3 power of weight. Because if you're converting volume to area, volume is a 3-dimensional term, area is a 2-dimensional term. If I want to convert a 3-dimensional term to a 2-dimensional term, you just take it to the 2/3 power. And these examples here are just two different scientists that took two different pathways. One was looking at within a species, what happens to heat production as you gain or lose weight. and the other across species. And those relationships are what we currently use when we're scaling information. Either we're thinking of it to the allometric scaling, which is 0.75, or body surface area scaling, which is 0.67. In chemotherapy, this was adopted by, in essence, by this paper published by Don Finkel, who is the first director of St. Jude's. And the rationale for this was based on this paper, was based on methotrexate and 3 other compounds. And here's the explanation for it. This table shows that we have different weights from 0.08 kilograms to, again, that 70 kilogram individual as the average. If I took the doses that were considered to be generally accepted doses and divided it by the weight, you get very different milligram per kilogram per day doses. However, if I take that same information and divide it by the body surface area across this really large magnitudes of size, you get roughly 3 milligram per meter squared. So the ability to sort of normalize this information across species was thought of as a simple way to sort of scale information, but also prevent potential overdoses in bigger individuals. So that's where body surface area became adopted. And Don Finkel in this calculation used the Me equation that I showed you before, and also used the Dubois equation. And the Dubois equation was based on 9 individuals. Some of you may know this already, but basically involved taking those 9 individuals, paper macheing them, taking the paper mache, calculating the surface area, and coming up with an equation. And this is the original Dubois equation. Many authors since then have said, that's kind of ridiculous. Why are we relying on 9 people? Maybe we need more people. So many people have done this. They said, we're going to take babies, we're going to take patients with cancer, patients without cancer, and come up with all kinds of different equations. And they've done this. And really, it was a New England Journal of Medicine article that basically, said, this is the way we do it, and it's simplified. It's known as the most stellar adaptation, which is in most textbooks. And I published a paper on this a few years ago. It's been a decade now, actually, showing that they're all essentially the same equation because they're all doing the same thing. They're taking surface area based on Euclidean geometry. So if I were to take any of these parameters as a 3-dimensional term and as a 1-dimensional term, and you sum them, you get the same answer. So it's the same scaling method, which is basically the square root. So just to show this in practical terms, if you take NHANES data and you look at what is body surface area for our population versus the weight of the population, this is what you get. And the relevance of it is, again, in terms of scaling. If I dosed someone between 40 kilograms, which is considered the bottom of the adult weight range, all the way to, let's say, 200 kilograms, I would be giving the 200 kilogram person 5 times the dose of the 40 kilogram individual, right? But if I were to use body surface area, I'm going to prevent myself from scaling that by no more than 3 times. So that's really all it's doing, is body surface area is preventing people from giving too big of a dose. So the risk when we're thinking about fixed doses is we're giving 500 milligrams across body weight in that 40 kilogram to 200 kilogram individual. The potential risk is treatment failure. For weight-based doses, because I might be giving too much, the risk is actually toxicity, but also could be financial toxicity, right? Like, can people actually afford these things? There's also a lot of just waste. There's been several articles showing that there's a lot of chemotherapy waste each year just because of this calculations. To solve this problem in the clinical setting, we use alternate weight descriptors like ideal body weight. I'll talk about that in a second. And then with body surface area, there's also a risk for treatment failure because you might actually underestimate the dose for some compounds. There's also risk for drug waste. And the risk here is people not understanding that body surface area is a nonlinear function of weight. And so I showed you those equations for body surface area. And sometimes pharmacists and physicians have used the wrong weight descriptor in those body surface area calculations and calculated the wrong dose or capped the dose unnecessarily. And so that's where the errors can body surface area dosing. So with weight-based dosing, we are risking, if I just took someone's weight and I dose them, I'm risking toxicity. To prevent that toxicity, we've come up with alternate weight descriptors. And when I sort of launched into this sort of career path during residency, one of the questions that came up on rounds was sort of, where does ideal body weight even come from? You know, it actually originates from University of Michigan. You know, so if you go in the hallways, look at 1974 and 1975, there's a faculty member named Benjamin Devine, and he published a paper, 1974, with this equation in it. That's basically 50 kilograms plus 2.3 times the height of individual over 5 feet. And did not cite where that came from, just published it, you know. And you can't believe how many people have cited that work or have included it in drug development since then. And so on residency in the 90s, you know, my mentor was saying, one of my mentors like, where did this equation come from? So I was like, there's got to be a reason. So that's how I was digging into it. I was looking at records from NASA, thinking, you know, there's got to be science behind this. And it wasn't the case. Actually, what it was is our love for the number 5. You know, it's kind of bizarre, but all it is really, if I convert this kilogram to pounds, right? It was just a rule. And the rule is that for every inch, you gain 5 pounds. That is the basis of that equation. And so I was able to find that in the nutrition literature as sort of a farmer's rule. I know Kathleen was up here doing the dosing. It's a very similar kind of concept. Now, if you were to use ideal body weight across a population, you would learn very quickly that we're now height-based dosing, right? Because this equation is just converting height into weight, which means that you're actually dosing people on their height. So you're going to underdose some people. And because of that, you have to correct it. And so that's where adjusted body weight comes from. It's the idea of like, hey, height-based dosing might not be good enough. You need to do better. Let's adjust it. So that's where that comes from. So really quickly, this is what you're doing with actual body weight dosing. If you were to use ideal body weight, you're going to get a height-based distribution of what the doses would look like. If you adjust that, that's the distribution. And then if you modeled this based on the principles that we use today, even at University of Michigan Hospital, this is what's done for some drugs. You use total body weight if it's less than the ideal body weight. You use ideal body weight if it's less than 1.25. And then you use adjusted body weight ultimately in most people because they tend to be over their ideal body weight. But if you fit that and you fit a power function to it, what you see is that it's actually the weight of the individual to 0.72. It's right in the middle of 0.67 paradigm to 0.75 paradigm. So we're doing the things that people figured out a long time ago, just calling it different things, right? So over the last 25 years, I'm not going to go through a lot of data. We've published a lot of papers, over 40 papers on this, but we've looked at this on a drug by drug basis, you know, just going after it. And what we've seen is that for some drugs, the way it has been approved is correct. But for a lot of drugs, it's not, it could be better. So we have drugs like vancomycin, daptomycin, as an example. Daptomycin is weight-based. There's ways to patch that error by using adjusted body weight. I believe fixed dose could be an option. Voriconazole is an example. That could be much better and was known from its development to maybe be genotype-based dosing. Let's figure out if this person's a fast metabolizer or a slow metabolizer and then decide the dosing. Here's a drug that we're working on now that we're really clearly showing should not be fixed dose, should be body surface area-based dosing. So there's many, many examples of this. But the point being, height and weight are really just what we've relied on for so many decades, right? And it's not good enough because we're doing so many things with that information. We're taking the height of an individual and the weight of an individual and making a lot of decisions, really important decisions. One of those decisions is even the classification of that individual as being obese or not being obese, right? So that definition is based on body mass index, which is the weight of the individual divided by the height in meters squared. So, you know, using this 1980s example of the twins, right? What you see is that you can have two individuals, Danny DeVito and Arnold Schwarzenegger, with really almost the same body mass index, but obviously very different body composition. So we're relying on the standard, which is BMI. By the way, that took about 150 years to get to resolution that that's the right standard. It's not perfect. It's just a good epidemiologic cutoff kind of value. And then in pharmacy, we've relied on ideal body weight. But ultimately, you can't see what's on the inside of a person, right? So you're looking at the individual. You can have individuals that look very similar, but actually have very different profiles. And often in males, this is a very common characteristic, which is known as the toffee phenotype, which is not an attractive name recognized for males, but it's like thin on the outside, fat on the inside sort of profile. So those diverse phenotypes have now been categorized many ways, and there's other subphenotypes of this. But the point is, again, body mass index is not a useful index sometimes when we're thinking about these kinds of features. And specifically, you can have individuals that have a lot of muscle mass. They're going to falsely be classified as being obese or overweight. You can have individuals that as they age are losing muscle mass. So as you age, once you get over the age of 45, 50 years old, you start losing muscle mass. And so you get a phenomenon known as sarcopenic obesity, but you can also have non-sarcopenic obesity, which is represented by these two figures here. So my work now, just transitioning to what I've been trying to work on, and will probably take most of my career, and hopefully I make a dent. It's going to be hard, changing height and weight as parameters, right? But key question, can we do better than these two parameters? And so the difference for me was really the Michigan difference. Coming to Michigan made a big difference. And one of those was when I came here, I had the opportunity of having, I met with a physician who introduced me to Stu Wong. And Stu Wong is a trauma surgeon. And over the last 15 to 20 years, has developed these algorithms really in collaboration with his son, who's a computer science person, Nick Wong, to develop these algorithms to help convert CT scans into body composition. And the major application of that has been in the auto industry to sort of figure out injury based on fat distribution and to support sort of where airbags should be placed in a car. So when I came in and I saw that, I approached Stu and I said, Stu, could we think about using some of this information for perhaps correlating this to drug dosing and exposures? And so that was a really fruitful discussion. And so the pitch today is really, can we find alternate scalars? And what I'm hoping to do with my work is move away from height and weight as those scalars to something what we're calling pharmacomorphomics, but really to address this fundamental problem that height and weight don't really tell us enough about the human phenotype. So what is analytic morphomics? Analytic morphomics relies on CT information. Most often that's through a lumbar CT scan or a thoracic CT scan. And the group has developed automated algorithms to take that CT scan information and then look at certain levels. What we found is that information at L3 and T12 typically give us really good information about the individual skeletal muscle mass. But there's many, many other applications. That group originally developed these methods really to characterize liver morphomics. So that's a space that we would like to work on further. But so far, we've been focused primarily on skeletal muscle area and some other metrics that I'll share with you. So with that, we've kind of published a series of papers. I'm not going to go too much into the detail of this, but we've shown that some of these pharmacomorphomic measures do have correlations to the clearance of a drug. Not so much the volume of distribution so far, but mainly to the clearance of the drug. We've shown this to be with these two key compounds, vancomycin and aminoglycosides, primarily because they undergo therapeutic drug monitoring. And because they undergo therapeutic drug monitoring, we can kind of leverage that real world data with available CT scan information. We've also done this with Methotrexate. Why did I go after methotrexate? A, because they do some therapeutic drug monitoring for the compound, and it's actually the basis of body surface area based dosing that I shared with you. So trying to kind of break away from those old paradigms requires evaluation of compounds that are established, right? And then through collaboration with Dan Hertz, we recently published a paper that showed a relationship between muscle mass and paclitaxel systemic exposure. And actually Dan is actually taking that to the next step, which is actually testing to see if we dosed using that parameter, would we do better? So that's an active ongoing study. So we've sort of leveraged our real world data to acquire federal support, and we've received through co-PI relationships 2 grants from AHRQ, and more recently a contract from the FDA. And I'm just going to give you the kind of the broad scope of those projects. So the first project is looking at surgical site infections. Obese individuals are higher risk for surgical site infections. We know that to be the case. The current standard is published in a guideline. That guideline says individuals that are over 120 kilograms should get 3 grams of cefazolin. If they're less than 120 kilograms, they should get 2 grams of cefazolin. So here's the funny part of this. So I look at this guideline and I see that recommendation and I see the citation. I'm like, where is this coming from? And I look at the citation and it's my paper. And it's a review article, you know? It's not original research. It's not based on anything. It's a review article that had a suggestion for a cutoff value for pragmatic reasons, you know? Because we think these cutoffs exist between 110, 130 kilograms. So they chose 120 and set that bar. So, you know, I could obviously leverage that and said, we can do better. And what we've proposed, so we're now at aim 3, but aim 1 was to look at the pharmacokinetics in patients. So we currently have our staff in the OR collecting, you know, basically fat samples, plasma samples with the support of anesthesiology. We attempted to do these individuals have their CT scans performed. We tried to find some pragmatic measures as well, because we recognize that there's going to be skeptics with our work as well. To say, hey, do I need a CT scan every time I'm going to dose someone? So we're looking for pragmatic measures like, let me use calipers, let me take a tape measure, let me get other metrics. Here was a surprise so far. Patients don't like getting pinched. Patients don't like standing up for tape measures. So ironically, the CT scans end up being better, in a sense, giving us more information than what we had thought of. So we did learn something so far. But the proposal aim 1 is to compare the morphomics to just standard measures, develop a pragmatic dosing strategy, and then pilot the effectiveness of the strategy. So what we've done so far is we've finished aim 1 and aim 2, which is we've built a mathematical model to understand what the best covariates are that predict the subcutaneous fat exposure of this compound. And then we've discovered sort of the pragmatic nature of what we can use through work. Shuhan is here today. He's helped lead a lot of this work. Has discovered that body depth is a useful metric for that. Work that had been conducted by the morphomics group prior to this had shown very clearly that subcutaneous fat thickness was associated with surgical site infections. So finding that this thickness makes a difference in the exposure of this compound is helpful, potentially. We also determined that kidney function plus this morphomic parameter helps us stratify this. And so we're now in aim 3 doing a prospective randomized study comparing the current standard of care versus this morphomic based dosing. And so that's undergoing now. Our second study that was funded is a lot more complicated, and I don't have a lot of data yet. And the reason for this complication is it's targeting patients with an appendectomy. What happens is patients that have an appendectomy can come into the emergency department and then have wait times of 18 plus hours. Most often what happens is elective surgeries have obviously specified times. Appendectomies occur when the OR is available. Often that OR could be available in the middle of the night or very early in the morning. So what ends up happening is a lot of patients are just waiting for the OR to be available. And so we put in a proposal to try and figure out the best way to figure out what those doses would be and what the prediction would be for mesenteric and this mesoappendix concentrations for antibiotics while they're waiting for the surgery to happen. As sort of this fail-safe approach to try and reduce the probability of surgical site infections. So this is ongoing because of that urgent emergent process, and because of we launched this during COVID, we're still at that 60% enrollment, and this is still ongoing. And then the third is a study that's being supported by the FDA. It's the same principles that I've outlined before. In this case, what we're doing is going to repurpose CT scans, and then we're going to have a prospective study component that validates that the morphomic parameter is better than weight for dosing this drug. Now, everything that I've said to you is about trying to get away from height and weight. But in principle, what is that? In principle, it's an approach to figuring out what that initial dose should be for any given person. How do we know it's the right dose a week later? How do we know it's the right dose a month later? It's just the empirical start, the beginning. So to get really precise, we believe that drug doses need to be adaptive. It should be adaptive either based on the response of the patient. So if you're treating high blood pressure, we can monitor blood pressure. If you're treating infectious diseases, that outcome might be too far away. We may not be able to determine when that's going to happen. And so we rely on concentration measurements. So typically our processes have been either measure a single concentration, use a lot of fancy math, figure out what the dose should be. If you've got more sample concentrations, you still probably have to use the fancy math. But we, in practice, often require on simple equations. The Sawchuk-Zasky method is one example of that, to optimize a dose of a compound. These are our typical ways. And the big problem with the system is that we have to collect samples, we have to assay them, and it's hard to do. So our group, one of our graduate students, Abby Lino, who, if you were on research day, saw her presentation, has been trying to overcome that barrier by looking at microsampling approaches, using at-home sampling, using two devices. In this case, it's the Tasso device. And what we've done is looked at this principle of volumetric absorptive sampling to support immunosuppressant dose optimization. And we've leveraged that more recently through work through the PK core to kind of leverage that measurement technology of using microsamples at 5, 10, 20 microliter dried blood volumetric samples for 2 pediatric studies. So there's one ongoing pediatric study that's funded and a second one that's has a fundable score. So we're waiting for the notice of award, but that is a multi-center study to help optimize antibiotic dosing in children. Shuhan is working on an obesity-related question. We recognize that, again, it's going to be very difficult to kind of sort out the dosing of every single drug that comes on the market. Can we figure out another strategy that will get us a better snapshot of drugs that are metabolized by four different pathways? Her study involves using 4 probes, microdose midazolam, caffeine with acetaminophen, and omeprazole, which allows us to probe these pathways. And then we're hoping that if we can figure out the system of what's happening in obese individuals, but not only obese individuals, but also obese individuals that are undergoing weight loss, that'll give us a much better model of what's happening to patients when their heights and weights are changing, so that we can then use that as a surrogate to optimize other drugs. So this 4-drug cocktail is in the works. Shuhan has been very successful at collecting samples. We're going to have our first abstract submission on this, and beautiful data so far. With midazolam, what we've shown is the opposite, counterintuitive. Obese patients have lower activity of CYP3A4 than the standard population. So completely unexpected. And in the first case that's gone through the weight loss, the individual who lost 27 pounds approximately, that person had an increase in their clearance of midazolam by 84%. So what we're showing is their clearance is lower. With weight loss, it comes back and it's back to baseline. So really important findings. And then Levi, this is a process in development. Just wanted to share really quickly. We were going to convert. What we're trying to do is get that estimate of kidney function using, again, CT scans. One of the things that happens with the CT scan is contrast agents are administered. A lot of those contrast agents are actually probes for kidney function. They're actually the gold standard probes for kidney function. So what we're trying to do is say, if someone has a CT scan and they have contrast, can we not only get morphomic information, but can we also get the true estimate of someone's kidney function? Because a lot of drugs, their dose optimization could be using that pathway. And then the last student, very quickly, is trying to answer another process, which is just this process of aging. I mentioned sarcopenic obesity, right? So you've got individuals who, after a certain age, lose muscle mass. There's many ways to characterize aging. Aging, we often think of as a covert on a chronological basis, right? So someone is 80 years old. However, that 80-year-old could be younger physiologically, or they could be a lot older physiologically. And there's a lot of now epigenetic tests to characterize that based on the degree of methylation, but there's also phenotypic methods to convert that. So there's a panel of 9 molecules that we could measure, like albumin, red blood cell size, cholesterol, and so on and so forth. Convert that to a phenotypic age. Epigenetic age could also be measured now. There's companies that sell these measurements for $300 per sample, as an example. So it's doable to get sort of the sense of biological age. So this is still in development. But just overall, sort of coming to the end of my presentation here, is that we want to optimize doses in specific populations. This has been the standard model, which is to learn and confirm. We want to do better. We want to take the information from the real world through that application process, use information that we have, which is available to us, and do better by better characterizing dosing in obesity, patients with organ dysfunction. We want to get away from race as a biomarker. We want better biomarkers than race, sorry. And we want to do better across that lifespan. So I want to end by thanking a lot of people. I want to start by thanking Dushin for giving me a chance. I'm not this traditional bioanalytical PK person. I sort of became that person in my journey. And Dushin really gave me a chance and really blew up my possibilities by making me part of a really big and wonderful team, which has been the PK core. I want to thank Bo and the whole analytical team who have helped generate a lot of the data that I kind of presented today, the graduate students for their work, and just all the staff that we have that kind of help make this happen. I also want to thank the Morphomics group because they also took a chance with me early on. They did all that work for me without any cost. They just, and it's worked to their benefit in a sense because we've been able to get funds to support that work, but it's really takes a lot of individuals to kind of generate that data. And then finally to the funders. So we've been lucky over the last couple of years to get funding. And I just want to also acknowledge industry partners over time who've helped support a lot of that work. And the group of individuals in Italy that have sort of developed really good relationship over the last 15 years who've shared real world data with me. In the United States, we don't do therapeutic drug monitoring that much for other drugs. We have the standard aminoglycoside vancomycin, but the groups in Italy routinely do therapeutic drug monitoring for a whole series of compounds. And so they've been very gracious in sharing their data, which I didn't talk about today, but has led to a lot of our publications. So thank you for your attention, and happy to take questions.
Thank you, Amit. So I have a quick question. How many of you online also know what Amit is doing this? That's much detail. That much not detail. So I think this is a really good opportunity. For me, I'm next door to him. I met him every week in 10 years. First time I heard a lot of things. First time I heard, honestly. So that's why I think if I met him every week, if I don't know what he's doing, people outside the university, in the university, would not know what you're doing. Fantastic work, right? So anyway, questions.
Amit, thank you for that incredible presentation. For those online, I'm Kathleen Stringer. I'm one of the disciples in clinical pharmacy. It's my chair. I'm trying to rack my brain. So pardon me if this is an ignorant question, but what really caught my attention was that you found that there is modifiable CYP3A activity independent of it being inhibited or modified by a drug, that there's physiologic modification based on body weight. Is that the first report of that? Are there other examples of that? Because
No,
that seems to be
so actually there've been
really,
that's a great question.
remarkable.
So just to, I think everyone heard your question. I think you were on mic, so
yeah,
I don't have to repeat it. But we are not the first to have reported this phenomenon. So there's 2 or 3 other research groups. The biggest one is in the Netherlands by Catharina Niebe, and she has published similar work showing that with weight loss associated with bariatric surgery, that you see an increase in the clearance of midazolam. And that's actually the first paper that showed this phenomenon. Where we're adding to this is information in the same individual because of diet and exercise associated weight loss. Often midazolam is used as a probe molecule using oral midazolam. What's unique about our model is we're using a microdose of 0.1 milligram of midazolam intravenously.
right. So that the change in clearance you can pretty confidently attribute to CYP3A activity.
Correct,
alone
because
versus not like improved kidney function, or I mean,
that's a great
you-
question. So the question is, how do we attribute that clearance just to that weight loss, right? And what we've done is we're actually measuring not just, I failed to mention this, we're measuring not just midazolam, but the major metabolite. So
Okay.
it's the ratio of that metabolite to the parent. And that activity is changing with that weight loss.
So you can attribute it to CYP3A activity, then,
Specifically.
not to changes in renal function or some other physiological phenomenon.
That's what we believe. That's what we're inferring. I don't know if I've covered all of the different potential possibilities,
Okay,
but that's the inference based on what we're seeing done in the literature is that
Okay.
that's the other isoenzyme systems, by the way, activity is higher in obese individuals. So 1A2, 2C19, that activity is higher in obese individuals, but 3A4 for some reason is lower. Why that is, I don't know. And I don't know if Haji has any insights into this, but one of the thoughts is like, we've got some individuals that have non-alcoholic NAFLD and NASH, and that's contributing in some form, but mechanistically how that happens, I don't know. What we know is that inflammation impacts CYP. So there's been hypotheses that obese individuals have chronic inflammation, and that contributes to this lower CYP activity. But
Tim have a question.
Hey, Amit, it's Tim Cernak in medicinal chemistry. Absolutely splendid talk. I love when you can kind of capture something from 1875 up to 2023. I feel like I learned a lot. As a medicinal chemist, I'm always thinking about like preclinical research. But I really wanted to hear your thoughts on microdosing and where, if that plays a role here. I'm curious about microdosing. When you showed that nice graphic of kind of very sensitive populations we test in, let's call it a standard human, but then you can imagine a pregnant woman or an infant or an elderly or someone with comorbidities. Can microdosing help to get that information in those more sensitive populations, or is that a preclinical person asking? We don't even need microdosing because it's already
No,
approved?
I think it's a great question. So this question of could we use microdosing as a strategy to get some information from individuals that I think I haven't seen that application necessarily happen, partially because there are concerns that the microdose might not be useful because we just would expect, we would not see the correct, we might just see a linear clearance phenomenon for most compounds, and it's not useful. We're using it to see changes in a given individual rather than trying to scale. What's been useful is in those kinds of specific populations that we consider vulnerable is reliance on physiology-based pharmacokinetic modeling. So there's modeling and simulation approaches to try and target those populations. So in the case, why we think this is a real phenomenon is there's actually an obesity PBPK model. Shuhan has actually run the midazolam analysis using that. And using that established model, we see that our data are very different from what the standard is currently. So that's the other reason. When we first saw this data, by the way, we were kind of surprised, skeptical, just like everything else. So Shuan and I couldn't understand what was going on here. So we ended up sending the samples to another lab. They confirmed the result, blinded. So we're sure now it's real.
And also the dose, the team just elaborate your question right now. Most likely the clinically FDA approved dose most likely are wrong, especially in oncology. Oncology has a big push today. At this moment, they're going to change the paradigm. So right now, the oncology is all MTD. Now they said, no, that's wrong. That's wrong because for many compounds, actually for PI3K kinase, other things, the suppress your cancer and the improve the patient progression-free survival. Then once we finish the phase 3 randomized trial, the overall patient survival is worse in treatment group. So FDA get this is wrong. So all the MPTD is going to change in next few years, going to completely change the dose regimen is likely wrong. You inhibit my tumor, you kill me. So will that help?
Yeah, great talk. I have a follow-up question related to midazolam PK. So you measure both parent compound concentration and metabolite concentration. So use the ratio as a sort of indicator of metabolism. So most likely CYP3A4 activity or CYP3A5 activity has changed in the patients. But also for these patients have a big change in body weight, right? So do you think that is also possible that the difference in PK is related to the difference in the disposition of the parent compound and midazolam in the body?
Yeah, I think it's possible. So, the question is, could there be some other explanations like changes in the volume of distribution of this compound and so on and so forth? And so for now, we only have it's preliminary, so I think our goal is to have 24 individuals and then see what's happening to them before and after. They're going to have various amounts of weight loss. So the example I gave you was the most extreme case of a 26 kilogram person. Most of them will probably have 10 or 15 kilogram change. But I think what would be important is to kind of see this not just with midazolam, but to see it with other potential probe molecules. I don't know if you have any. There have been mixed things that I've seen in the literature to use, like, for example, 4-beta hydroxy cholesterol or some other marker. Do you have any thoughts on like?
Yeah, some people use some endogenous 3F4 substrate as a kind of biomarker. But yeah, there's some complicated results, let's say.
Yeah,
So-
and as we've been thinking about like what else could we use to kind of validate that this is a phenomenon, not just a midazolam thing, could it be something else? We've tried to look for other substrates. Unfortunately, there's not a lot of options.
Thank you.
You had a slide with a single table where you had maybe 10 or 12 different drugs and what the recommended dosing was and what you guys recommended and said. I was surprised to see some examples there where it was weight-based dosing and you recommended fixed dosing. When you are doing a project and you come up with a recommendation to say it's fixed dosing, is that based on there not being an association between the body size and the resulting kinetics, or because there's something about those drugs that the kinetics don't matter to the response? Is that more of a PK/PD exposure
Yeah,
response that leads you to make that determination?
So the one example that I recall from that table, I'm not going to pull it up now, is a drug called telavancin and daptomycin. Telavancin is an antibiotic that has a very, there are things about that molecule that were known preclinical development that said it was a fixed dose drug. But the company went along. And I'll give you this example because it's firsthand, because I was through that process. So telavancin has a very small volume of distribution compound. They're typically in that 0.1 liter per kilogram range. What that means when you see molecules with that kind of profile, it means they tend to stay in the plasma compartment. They don't go so much anywhere else. What that also means is that that kind of compound is not going to not only not distribute to other tissues as well, but that the scaling of that is not going to follow a linear with weight, right? The challenge with that molecule was that it's a glycolipopeptide. It's associated with kidney injury. And in animal models, they showed very clearly that if we gave higher doses, we would see toxicity. So there was a clear risk. The company proceeded with a weight-based dosing regimen. And soon after the drug was approved, there were publications showing that overweight individuals had higher kidney injury than non-obese individuals. When that was published, I wrote a letter to the editor, took their population PK model and showed it's obvious. Bigger people are going to have higher exposure from your model. They should get lower doses. The company fought me on this, of course, right? So that's 2007. 2012 is when we published that letter. It took another 7 years to get the money to do a fixed dose study. And we finished that study. We published it in 2018, 2019. We showed if you gave different cohorts 500 milligrams, 750 milligrams, or 1,000 milligrams fixed dose, you actually still see an increase in exposure. So that 750 milligram dose actually worked for the whole group. But the drug has already been on the market. No one's using the drug. The life cycle has ended. No one cares, right? So that's that, except for me. So it's like, that's the challenge, is like, how do we get people to see early on? And it's like, we have to get it literally at phase 2. And often that's proprietary information. You can't.
Technologies. You,
Yeah.
from the very beginning, those, but today, that's a huge difference. Everybody changed, right? That's a good example. Everybody gains.
Right. Tacrolimus is another example of a drug that's on a weight basis. Probably doesn't need to be weight-based.
So, anybody have question online? Oh, there's a question online. Q&A. I said, I care still. Love it.
Okay, thanks, Sam.
Thank you. Yeah. Thank you. With that, thank you very much. With that lovely note, thank you very much for attending the seminar, and see you next time. Next time is April 28th. Thank you.
Okay.